I want you to imagine a medicine that is so personalized, it's designed and manufactured for just one single patient. Not a group, not a clinical trial, but for one person. Now, this isn't science fiction. This is a real-life medical story that could completely change how we think about curing genetic diseases. So, let's dive into how it all happened. Right. This wasn't some off-the-shelf drug. This was a completely bespoke therapy, engineered from the ground up to fix a deadly flaw in one person's DNA. In the medical world, they have a term for this, an N of 1 therapy. It means the number of patients, N, is literally just one.
For this particular patient, the clock started ticking pretty much the moment he was born. Within just 48 hours, this newborn baby boy was in critical condition. Yeah, instead of being alert and awake, he was lethargic, he was struggling to breathe. All of these terrifying signs that something was desperately, desperately wrong. For a tiny newborn, you can imagine the stakes just could not have been any higher. So, this first part of the story is really all about that initial crisis. It was a frantic race against the clock to figure out what was happening and, well, to save his life.
The diagnosis came back as CPS1 deficiency. Okay, so what does that actually mean? Well, think of it this way. The body's waste disposal system for protein is just broken. A substance called ammonia, which is incredibly toxic to the brain, can't get cleared out, and it just builds up to deadly levels.
And that is exactly what was happening here. The patient's ammonia level had just skyrocketed to over 1,000. Now, to put that in perspective for you, the normal safe range is somewhere between 9 and 33. This wasn't just a little high. This was a catastrophic, life-threatening crisis. And here was the awful dilemma. A disease like this has a 50% mortality rate in early infancy. Every single moment those ammonia levels are high, you risk permanent brain damage. Now, the only real cure is a liver transplant, but an infant this small is simply too fragile for that kind of surgery. They were completely stuck in this terrible waiting game. So, with the patient stabilized for the moment, the big question became, what was the root cause of this devastating disease? Well, the answer was found buried deep inside his genetic code. It's almost unbelievable when you think about it. All of this chaos, this life or death situation, was caused by a single typo in the 3 billion letters of his DNA. Just one wrong letter created a premature stop signal, which meant the crucial CPS1 protein was never fully built. And just like that, his whole urea cycle was useless. But identifying the problem is one thing, fixing it is another challenge entirely. And this is where a truly revolutionary technology offered a sliver of hope. So we've all heard of CRISPR, right? It's often described as genetic scissors that can cut DNA. But the technology used here, called base editing, is way more precise. And this analogy is just perfect. It doesn't cut the DNA. Instead, it chemically rewrites a single letter. It really is like using a pencil and an eraser to fix one tiny typo in a giant book. And the process itself is just incredibly elegant. First, a guide RNA acts like a GPS, leading the editor to that exact typo. Then it locks on, and a specialized enzyme, that's our pencil and eraser, performs a quick chemical swap, rewriting the faulty A to a healthy G. And finally, it just lets go, leaving behind the corrected gene, ready to do its job properly. So the science was there. The tool existed. But could a team of researchers actually design, create, test, and get regulatory approval for a brand new drug for one specific person, all while he was fighting for his life? The sheer speed of this whole process is what's truly mind-boggling. Just look at this timeline. In just 6 months, the team went from a diagnosis to a fully approved, customized gene editing therapy. They developed cell lines, found the perfect editor, ran safety studies, and got the FDA to approve it. In the world of drug development, this kind of pace is absolutely breathtaking. So this brings us to the most important question of all, right? After this incredible sprint of science and medicine, did it actually work? Well, the data here really speaks for itself. Before the therapy, his median ammonia level, even with all the intense medical care he was getting, was 23. After the first infusion, it dropped to just 9, smack in the middle of the normal range. After a second dose, it stabilized around 13. This was the first clear sign that, yes, the therapy was working. And that biological effect translated into a real-world clinical benefit. The patient's daily dose of nitrogen scavenger medication that's the drug that was helping control his ammonia, was successfully cut in half. That is a massive improvement in his quality of life.
But it gets even better. He could actually eat more protein, which is so crucial for a growing baby. When he got sick with common viruses, things that before would have triggered a life-threatening ammonia crisis, he recovered without one. And so importantly, he was growing, he was gaining weight, and his neurological status stayed stable. He was just more resilient.
The success in this one patient is just incredible in and of itself, but the true significance of this story really lies in what it means for the future of medicine.
And this is what makes this approach a potential game changer. It's less like creating a new drug from scratch every single time and more like a platform. The delivery vehicle, the base editor itself, those can be standardized. The only part that needs to be custom-made is that guide RNA, the little GPS molecule. This could make it possible to rapidly create therapies for thousands of other rare genetic diseases. Oh, and plus, you can re-administer it if needed, which is a huge advantage over many other gene therapies out there.
Look, this case was a proof of concept. It was a stunning demonstration of what is now possible. And while there are still so many challenges ahead, it forces us to ask a really profound question. Are we witnessing the dawn of a new era of truly personalized medicine where we can create a unique cure for every unique disease?